Okay, hello to all of you, and welcome to this free talk series on an introduction to nucleic acid therapeutics. My name is Owen Dunhu, and I hope you're all doing very well. Over the course of these talks, I hope you can gain some kind of an insight into the potential clinical significance of nucleic acid therapeutics, their modes of action in vivo, and the next generation of nucleic acid therapeutics that are now well and truly entering many R&D pipelines. So this will just take the form of three talks. As indicated in the opening slide, we'll start today with going through the clinical significance of nucleic acid therapy. And in fact, this will be split up over 2 weeks. First, this week, we'll talk about the different modalities with some examples, mainly focusing on nucleic acid therapy against some inherited diseases, some rare and some not so rare. And in part two next week, we'll talk about nucleic acid therapies in cancer, as it's an important emerging area, and I think it really deserves its own lecture, as I'd like to introduce a small bit, but not too much, cancer immunology as well, which I think would be really beneficial to provide some context to help understand the importance of nucleic acid therapies against cancer. Then finally, we'll look at the next generation of nucleic acid therapies on the last week so that you will get an idea of where this field is heading. And there are several things we'll focus on here that you'll be hearing more and more about over the next few years. And there's some genuinely exciting developments and technology emerging, and it's a very interesting time to be in this space. So I'm looking forward to sharing this with you later on in these talks. But we'll get started with today's talk, which will be an introduction to the clinical significance of nucleic acid therapies. And we'll start with what it is that distinguishes nucleic acid therapeutic molecules from other conventional protein based biopharmaceuticals. And essentially, what you're doing here is you're using nucleic acids that are manufactured outside the cell to alter what proteins are produced inside the cell in a way that is in some way beneficial for patients. And the basis of all this goes back to that central dogma of molecular biology, which I'm sure you're probably all very familiar with. But briefly, as you know, the instructions for building proteins, which are called genes, are encoded in certain regions of DNA strands, which are replicated during cell division and inherited by all subsequent descendant cells. And for these instructions to be read, this information needs to be transcribed into another format called RNA first. And once it's in this format, it can be translated into proteins by the cell. So if you introduced the DNA based instructions for a protein of interest into a cell and got it into the nucleus, you will get that protein being produced by the cell, which can either then stay in the cell or be secreted out of the cell and get around to other parts of the body. And of course, you don't need to always introduce DNA. Nowadays, you can just as easily introduce RNA, which just needs to get into the cytoplasm. And if you can do this, you'll have the same results. With one key difference, of course, in that with DNA, the alteration of the DNA content of the nucleus leads to permanent or at least long term changes in the recipient cells. But the introduction of RNA, on the other hand, leads to an extremely short term change. And this is because the RNA degrades very quickly, unlike DNA, which can last in the nucleus indefinitely, essentially as long as the cell survives. So the effects of RNA compared to DNA are very transient, but it's enough in some cases, like with vaccines. But in other situations, in order to engage in successful long term treatment of conditions using RNA, you do need continuous doses of RNA. So, RNA may not be unlike normal biopharmaceuticals in that respect. But also, in some cases, unlike DNA, it's this transient impact that makes it safer. In the case of any adverse responses with proteins that are produced from the RNA, this can be counteracted to presumably a large degree by simply discontinuing the treatment. And how long it takes for the adverse effects to stop then would largely be dependent on the stability of the protein of interest or the other impacts of the RNA treatment, because as you probably know already, not all RNA produces proteins, but we'll talk a bit more about this later. But of course, having the option to discontinue treatment as a first step is really useful. But of course, this is not the case with DNA treatment. DNA treatment is typically designed to be used once, and the impact, good or bad, will last, especially if it involves any kind of changes to the genome. So, RNA, as well as from an efficacy standpoint, is also quite attractive from a safety and regulatory standpoint. So, although DNA based nucleic acid therapy has been with us for many years, recently there's been a lot more interest in RNA in the therapeutic space. So, we'll focus on this first. So, what is different about RNA and to some extent DNA as well, let's not forget, as a therapeutic molecule, is that it doesn't need to be as expensive as traditional protein based biopharmaceuticals to produce. And this is because the routine upstream processes are less challenging, typically involving either a chemical process such as solid phase synthesis in the case of small RNAs, or in the case of longer RNAs such as messenger RNAs, this involves enzymatic processes such as in vitro transcription. But in both cases, it's actually really easy to accomplish. And many of you will probably have seen this process summary before elsewhere. And what's good about this process here with the manufacture of RNA is that you don't need bioreactors or fermentations with each batch. So once you get to this point here and you have a large stock of your plasmid, which can be used as template for in vitro transcription, in many cases you'll have enough plasmid for multiple batches or possibly enough plasmid to do you many years. So for some perspective, in our lab, we can generate about 30 micrograms of purified RNA in about 3 hours with about 15 minutes of hands-on time using 1 microgram of linearized plasmid. And this costs us an estimated €20 to do in terms of consumables. But our process, of course, isn't very efficient, and if you scale this up and make it more efficient, the price of producing RNA plummets. And this is true for solid phase synthesis too, for small RNAs. And because there's not any living material here within these production systems, and indeed the systems are not designed to support life, there certainly are less sterility issues with these processes. And also because you're not relying on living organisms, batch to batch variability, or long term stability of your cells or bacteria is not something that's as much of an issue. Even in terms of downstream processing, what you're starting off with after your RNA production are very simple mixtures that your end product needs to be purified from, with only one or two contaminating proteins and some other components. Also, from one mRNA product to the next, the physiochemical properties are more or less going to be the same, unlike protein based biopharmaceuticals. So there's minimum adaptation required for new products, of course, making process development and validation easier between different products. So essentially, if producing DNA or RNA based biopharmaceuticals, rather than going to all the trouble of producing a protein in a factory, making sure you have the correct glycosylation patterns and potency, etc., if you just produce the nucleic acid for these proteins in a factory, the cells in the patient will take care of the protein production. It will do its own QC and batch release. It's much better at this than us after billions of years of fine tuning. Our cells are very good at these things, and it's what keeps us alive. On the other hand, it's important to remember that mRNA or DNA based treatment is not the answer to everything. In many cases, treatment with protein based biopharma products is still preferable regardless of the cost. And at the end of the day, the shortest and most effective way to get a therapeutic protein to the right place in sufficient quantities to be of any therapeutic use is to simply put the protein there. But really, it depends on the application. For example, looking at RNA first, in the case of the COVID-19 vaccines, the quantities of spike protein production required in vivo were not huge. As lucky for us all, you only need a small amount of foreign protein production in your body and appropriate immune stimulation in order to develop a long term adaptive immune response against most non self proteins. But in the context of therapeutic intervention, you may need, for example, up to 1,000 times more protein production from mRNA in order to reach a therapeutic threshold, or maybe even more if it's a secreted protein that needs to enter the bloodstream for systemic delivery and impact. So, in addition to getting RNA to the right places in the body, you also have to ensure that you get adequate protein production. Also, given that mRNA or its delivery vehicle can stimulate an immune reaction, this is something that may, of course, impact protein translation in cells. And also, because of this immune stimulation, using mRNA as an alternative to recombinant protein may be problematic in some circumstances. For example, if you need several injections per week, as is the case with the usual administration of recombinant factor 8 and factor 9 for hemophilia A and B patients, for example. So it's very important to keep this in mind. And this brings us back to DNA based therapy, which is much better for sustained long term expression of therapeutic protein, avoiding or reducing the need for repeat doses. But here, of course, the production of DNA based nucleic acid therapy products is not as straightforward as RNA, principally as its delivery requirements are much more specialized, as it needs to get to the nucleus. And this is typically best achieved by packaging it into viral vectors, which will survive in the extracellular environment and easily enter the cell and deliver the DNA to the nucleus, where the gene can be transcribed into RNA and then translated into proteins. And these are generally non integrating viruses, so in theory, the DNA will stay doing this in the cell nucleus indefinitely, as long as the cell continues to stay alive. Hence, the more sustained impact of DNA based therapy. But the production of these or any virus based products are really not as straightforward as mRNA. And I used to work in a company producing an inactivated viral product. And the sheer amount of work and really good organization and management that goes into producing one batch of viral product in the real world is absolutely enormous. And this is something that we don't really always appreciate when we're learning about these things in university before we start our careers in industry. So for DNA based therapy, there's a lot more involved in its production. And also, as mentioned earlier, there are safety issues here. The impacts of DNA based therapy, good or bad, are long term or permanent. So the R&D and clinical processes for these are much more difficult. But one of the key things to remember about mRNA and DNA based therapy is that it can do something that recombinant protein based therapy can't do so well, which is it can act from within the cell. Recombinant proteins usually can't do this. They're usually restricted to acting outside the cell, as they have an ability, for example, to cross the plasma membrane. So they mainly work via extracellular interactions, unless they can be potentially fused to something else which can be taken in by the cells. But this is not going to be possible in all cases, as such fusions may impair the function of the protein of interest. So there are many genetic conditions that can't be treated by simply administering large amounts of protein based biopharmaceuticals. This is particularly true for metabolic conditions that lead to a toxic buildup of biomolecules that unfortunately lead to some very difficult and chronic and life threatening conditions, such as the examples listed here. These kinds of conditions really need to be tackled from inside the cell so that you're treating the problem at source and not just treating the symptom. Similarly, in other cases where you have genetic conditions where you need to replace cell membrane proteins acting as receptors or transporters, these proteins need to be made within the cell first and then transported to the membrane in a natural and regulated way. And you're typically not going to be able to produce proteins in a factory that will do this for themselves for every disease. And a good example of this problem is with the replacement of the CFTR cell membrane protein in order to counteract cystic fibrosis. And this is something that I'll talk about in a bit more detail later. So, by delivering the correct DNA or mRNA, we are giving cells the information they need to make beneficial changes from within, because there are many medical conditions where we simply can't achieve this from the outside. So while DNA and mRNA based therapies are not perfect in terms of being a solution in all cases, and there are many technical barriers to overcome, the progress that's been made here in this area in the last 5 years has been huge. And of course, this has been kickstarted in a huge way by the COVID-19 mRNA vaccines. So this long held idea of changing the cell from within is now feasible, and opens up the door to treat so many more conditions that need to be tackled from within the cell. And if you learn anything from today's talk, I hope this will be it. This is really the key point behind nucleic acid based therapy. It's really in this context that nucleic acid therapies have received a lot of interest, and particularly RNA, because its impacts, as mentioned, are not permanent. Treatment can be discontinued easily, making it much less risky. But of course, translation of genes delivered by DNA or mRNA within cells can also be used to secrete proteins out of the cell, getting proteins to the bloodstream and achieving systemic delivery of therapeutic proteins. So, if harnessed correctly, nucleic acid therapy doesn't have to be restricted to just inside cells. It can have an impact outside cells too, just like recombinant proteins. So, you get the best of both worlds here. But as I alluded to earlier, there needs to be enough protein produced in order to get to that therapeutic threshold. So, for example, either DNA or mRNA therapy has been used in the production of antiviral antibodies, immunomodulators such as IL-2 to treat autoimmune disorders. And these are good examples of a systemic delivery of proteins through nucleic acid therapy that have got to at least stage 1 clinical trials. Nucleic acid therapy can also be used to secrete factors that also work at a much more local level, such as DNA or mRNA delivered VEGF-A to counteract heart disease. And these are good examples of nucleic acid based therapy against non inherited conditions. So there is no protein replacement happening here. It's an introduction of new proteins, like in the case of an antibody, or the supplementation of existing proteins. So that's a very general introduction to the clinical significance of nucleic acid therapies and where they might fit in the future of medical care. But it's a very broad area, and when I sat down to try summarize it in some way, in some kind of, I guess, a systematic way, um, that might be easier for you to digest, it was kind of difficult to know where to begin and where to end. And some aspects of it are hard to categorize. But what I ended up with was this table here, which I think is a decent overview of the main areas. And there's a lot of text here, so I'm not going to go through it. It just summarizes what we'll be talking about for the rest of the lecture. And I'll provide this file for you as well, so you can browse it yourself at your own leisure. You can use it as a very quick reference guide. So in the introduction, I mentioned nucleic acid therapy in the context of protein replacement or supplementation therapy, as it's similar in principle to protein-based therapy that we're more familiar with. But as you can see from the table, nucleic acid therapy is far more than just protein replacement. Nucleic acids can be used to conduct targeted gene silencing or mRNA altering, which you were introduced to in earlier lectures, or combinations of gene silencing and protein replacement. But also, as some of you might know, nucleic acids can also be used to make changes to genomes and cells, for example, through the use of CRISPR-Cas9 systems, which largely depend on the insertion or deletion of bases in a genome through the use of double-stranded breaks. And this is something that I'll go through in a little bit more detail later on in the lecture. There are also some emerging gene editing approaches that can make direct changes to genetic code without insertions or deletions or any double-stranded breaks. And these are really exciting, but I'll probably talk about these on a different day under the category of emerging technology. Also featured on the list here is the use of RNA in ligand binding. So maybe not many of you would have known this, but RNA molecules referred to as aptamers, which are essentially like pieces of RNA that fold into secondary structures, can actually also act a little bit like monoclonal antibodies. And you can develop them without relying on the immune system of animals, and they have much smaller structures than antibodies, which is a huge advantage. And I'll talk a little bit more about them in a different lecture as well, but maybe not today. And as you can probably guess, most of these processes can be achieved using either RNA or DNA or combinations of both, as you can see from the different coloring systems here, with RNA possibly being more versatile, and of course the impact of RNA is short term, except for when it's used to make changes to genomes and cells. But it's the transient action of the RNA in this context. In this context, is still quite useful, and we'll learn more about this later too. So, a lot of nucleic acid therapy is used to counteract or correct inherited genetic conditions. But which approach we use really depends on the underlying biology of each condition. But all genetic conditions, regardless of their biology, are caused by mutations. And this is something that you're all going to know already, of course. But for the purpose of being brief, we'll just revisit it here again, and we'll stick to mutations in protein coding genes, because of course mutations can also occur in non-coding genes too. But we'll just look at coding genes today. So as you know, in protein coding genes, the genetic code is read 3 bases at a time in units called codons, with each codon being translated into a different amino acid. And this dictates the order in which amino acids are combined together by the ribosome to construct proteins. But sometimes you get mistakes occurring during genome replication, which occurs during cell division, and the codons can change. And sometimes this results in no actual change to the amino acid, as sometimes the same amino acid can be represented by multiple codons. But sometimes the change in the codon does lead to amino acid change, and this may have an impact on the function of the resulting protein. Furthermore, as well as these more simple changes, you can also have base deletions or insertions. These can be much more detrimental. For example, a one base pair deletion in a codon also impacts the codon after it, as they are still read in groups of three. So, by way of a simple analogy, imagine if we just read sentences 3 characters at a time with a space every 3 letters, no matter what. Deletions and insertions would also be a problem in this system. For example, look at this simple sentence here. If you were to delete the first E, you get this, which makes no sense, as your reading frame is shifted 1 character to the left. Well, this is exactly what happens if a deletion or an insertion of a base occurs in a protein coding gene, and it's called a frame shift. And its impact on protein sequence depends on whereabouts in the protein sequence that it occurs. Just like the examples here with the sentence, insertions or deletions have varying degrees of impact on the preservation of the sentence meaning, depending on where they occur. And it's the consequence of these mutations, in other words, what the mutated protein does, that really dictates what kind of nucleic acid therapeutic approaches are suitable and not suitable. So it's not an ideal situation if mutations happen, of course, but if they do, the best scenario is where the mutated protein does absolutely nothing at all. And this is because most of the time in these cases, this is largely okay, because as humans, we have 2 copies of each gene, one from each parent. So in most cases, we'll have a backup copy of a gene that produces enough working protein that's well over a critical threshold. So there's really no bad effects in a lot of cases where we have a mutation in one of our genes. So this is a classic example of what we refer to as a potential recessive disease causing gene, which means you can actually have a gene that causes such a disease in your cells, but your cells can produce this non-functional protein, but it doesn't really matter, as it doesn't do anything, and there's plenty of normal protein there to compensate for it. So such individuals are referred to as being heterozygous for this gene. In other words, having different versions of the gene, one good and one bad in this case, as opposed to being homozygous, which is having 2 good copies. In which case you have even more functional protein, and you are essentially a non-carrier of the disease causing gene. But you can also have the opposite, of course. In other words, you can be homozygous with 2 non-functional copies. And with recessive disease causing genes, it's only in these situations where you develop a disease, simply because you only have non-functional versions of important proteins with no real backup. And a good example of a recessive disease causing gene is the gene for cystic fibrosis, which I mentioned earlier. And it's caused by various mutations in a gene coding for the CFTR protein, which is embedded in the cell surface in the lungs and elsewhere. And it's important as it's needed to be able to pump water and ions out of the cell. because in the absence of this process, you get abnormally thick mucus in the airways, leading to many complications. As it's a recessive gene, heterozygous carriers are unimpacted by it. There's still plenty of functional protein in these carriers, pumping water and ions out of the cells, so you don't get the same complications. It's only when people inherit two copies of the mutated gene and are homozygous carriers of the gene that this process is entirely absent and they then present with the symptoms of cystic fibrosis. So, in terms of therapy, this is a classic example of a condition that requires a protein replacement approach, where the change must come from within the cell. So, in terms of therapy, the ideal solution would be to get the correct genetic information into the cell using either RNA or DNA, and to get the cell to produce enough functioning CFTR protein, shuttle it from within the cell in a regulated way, and get it to be properly embedded in the cell surface, where it can then begin to engage in the transport of water and ions out of the cell and begin to counteract the complications that are caused by cystic fibrosis. And although this has been tried with DNA in the past, since the 90s, it wasn't very successful. And more recently, a company called Translate Bio have been trialing a CFTR mRNA approach, with the mRNA being delivered via an inhaler. And while they have been able to demonstrate safety with this, the initial results from the clinical trials phase 1 were a bit underwhelming. And from looking at the publicly available data, there didn't seem to be that much of an immune response to the treatment, which is good in this case. It's you want to keep it down as it impacts on protein production. But one possibility for the underwhelming results here could be due to their attempts to broaden the recruitment to the trial, as they did accept patients that were also receiving other reasonably effective therapies, which alone would have boosted the FEV1 levels, which was their primary readout. And it might have really masked the true effect of the CFTR mRNA treatment on the FEV1 levels. But also, the fact that the study was statistically underpowered probably didn't help. But to be fair, the primary purpose here was to demonstrate safety, not efficacy. But the trials are continuing, and we'll see what happens. Maybe they will review their patient recruitment patterns for the next phase. In parallel, though, Moderna and Vertex are also collaborating on trials to test a similar mRNA that's also inhaled, but there's not as much information available publicly about this at the moment. But they'll certainly be interesting to keep an eye on. So, protein replacement is ideal for conditions caused by recessive genes, but there are also medical conditions that are caused by dominant genes. And in this case, you just have to have one copy of a disease causing gene to result in the development of a genetic medical condition. And in its simplest form, this occurs due to what is referred to as haploinsufficiency. Typically, each protein needs to be produced at a certain level in cells for normal physiological functions to proceed without the development of medical condition. So sometimes one copy or one functioning copy of a gene is more than sufficient to ensure that there's enough of a particular protein being produced in cells. But for other proteins, then, both copies of the gene need to be present and functional in order to ensure that there's enough protein being produced. So if one of them becomes non-functional due to a loss of function mutation, the remaining copy isn't sufficient to produce enough functional protein. So this situation is what we refer to as haploinsufficiency, and it applies to about 15% of human genes. So you can be heterozygous and have a functional protein that's still being produced, but there just isn't enough of it being produced from a single copy of a gene. And it's this deficiency then that leads to the development of some medical conditions. So this is a bit different to the recessive conditions that we talked about earlier. In this case, the condition is dominant. You only need to have one copy of the gene, as I said, to end up with a condition. And in theory, you can also be homozygous for these kinds of mutations, having a loss of function in both copies of a gene, leading to a much more severe outcome. So, a good example of a gene that exhibits haploinsufficiency is the gene for the cell surface receptor called LDLR, which is responsible for removing LDL cholesterol from the bloodstream, and this mainly happens in the liver. But the loss of one copy of the LDLR gene due to a loss of function mutation results in a condition known as familial hypercholesterolemia, or FH for short. So heterozygous carriers of these loss of function mutations don't get enough cholesterol removed from their blood, as there's simply not enough functional LDLR to do this, which increases the risk of heart disease. And notably, approximately 85% of individuals that carry one copy of a non-functional LDR gene go undiagnosed. So it's understandable why you might end up with people being born with two copies of this bad version of the LDLR gene. Again, this is an ideal candidate for protein replacement via DNA or mRNA treatment. And in preclinical trials, the delivery of functional LDLR via mRNA has worked really well at reducing LDL levels in mice. And these are mice that are homozygous for a non-functional version of LDLR. So, like this example in the bottom here. And this mRNA based approach towards treating FH has got as far as phase 1 clinical trials, but there hasn't really been any update on the results yet. But overall, there hasn't been a huge movement towards LDLR replacement using mRNA as a means of treating FH. And this may be because there's already some approved gene silencing drugs out there that can tackle the problem in a more indirect manner by targeting another protein in a way that can benefit individuals that have a non functional LDLR gene. And we'll talk about this next. But it might be interesting to watch this space over the next few years, as I think that a more direct approach of delivering the LDLR gene to the cell in the form of nucleic acid may be even more beneficial, alone or in conjunction with these existing therapies. Overall, these loss of function mutants that are either recessive or dominant if there is haploinsufficiency, it's because the mutant protein does nothing that it's possible to just supply the correct protein. And this is the simplest or least risky form of nucleic acid therapy because you're delivering an mRNA for a protein that should already exist in the body. So the action of these translated proteins are less likely to result in adverse effects. So in some sense, diseases caused by loss of function mutations, especially recessive ones, are the low-hanging fruit of targets for nucleic acid therapy. And I say this in a positive way. With genetic diseases, the more low-hanging fruit we have, the better for sure. These are the types of conditions where the simple introduction of the correct nucleic acid is something that's more likely to work. And this is reflected in lots of R&D pipelines. For example, if you follow Moderna's progress, you'll see that all of the diseases that they're targeting with their therapeutic mRNA can all be caused by loss of function recessive genes. And also, they're all rare diseases, so some may qualify for orphan drug status as well from regulators, which would be a huge boost to these clinical programs. But in addition to these dominant mutations that are caused by loss of function mutations in one copy of a gene, where you simply end up with a disease because the mutant protein does nothing and there's not enough functional protein being produced, there are also other types of mutations where the protein can actually gain an unwanted function. And this unwanted function may be toxic to the cell or the body. And in these cases, you may still have one normal functioning copy of a gene. There may even be enough of this protein being produced, but it won't matter if the mutated gene produces a toxic protein. There's also dominant negative mutations that lead to other proteins or other mutant proteins, that actively inhibit the function of the good protein. So in both of these scenarios, having one copy of a gene leads to a disease, no matter what. So a simple protein replacement is not going to work in these cases because it doesn't really resolve the problems caused by a gain of function or dominant negative mutations that are still going to be present. What may work better in these cases is gene silencing. And this is something that you would have been introduced to in earlier lectures. As you can probably recall, gene silencing can be done through the use of antisense oligonucleotides that are just small nucleic acid molecules that are complementary to target messenger RNA transcripts. In other words, they just stick to them through complementary base pairing. So by just changing the sequence of the bases, you can program these to target, in theory, any mRNA, and they represent the first truly programmable form of drugs. And as you know, they can act through molecules such as siRNAs. And as you also know from earlier lectures, siRNAs are small double-stranded RNA molecules that work through a process called RNA interference, or RNAi for short. So RNAi is a naturally occurring process used by cells and even viruses to silence messenger RNA through the use of natural siRNAs called microRNAs, or miRNAs for short. And it's an area that I'm particularly interested in. Many years ago, I discovered a new viral miRNA, but for sure the field has moved on a lot since then, and it's nice to see RNA interference now being used in the clinic. And siRNAs, as you can see here, work by essentially mimicking these natural uh, microRNAs being incorporated into the RNA-induced silencing complex, or RISC for short, and then guiding this mRNA suppressor machinery towards target mRNA for cleavage. And this is a little different to the typical action of microRNAs, which is their natural counterpart. And here's a simplified version of events depicting more or less the same process. And underneath here we have another type of antisense oligonucleotide referred to as gapmers. These are similar to small interfering RNAs, but they are delivered as a single-stranded molecule, and they're made of RNA with a DNA core. So they don't get incorporated into the RNA-induced silencing complex like siRNAs or miRNAs. Instead, they bind to mRNA themselves, creating an RNA-DNA hybrid. And the presence of this activates cellular RNase H systems, causing it to degrade the target mRNA. And unlike siRNAs, they can operate in the nucleus too, as opposed to just operating in the cytoplasm. And as a good example of a genetic condition that can be solved by gene silencing, we can return to FH. As in addition to mutations in the LDLR gene, which I mentioned earlier, some people can have mutations in the PCSK9 gene that also leads to the same condition. So this gene encodes a protein that naturally plays a role in the degradation of LDLR on the cell surface, which is an important natural process too. So if you have two normal copies of this gene, you'll be fine. You'll have normal background levels of LDLR on the cell surface, and your blood cholesterol should also be fine. But if you have just one copy of this overactive gain of function mutated version of this protein, you'll get too much LDLR degradation on the cell surface, leading to FH also. It doesn't really matter if you're producing enough normal PCSK9. The presence or absence of the normal version here is irrelevant if you have a gain of function mutant that is essentially toxic. So this is a classic example of a gene that needs to be silenced. And this can be done by using an siRNA called inclisiran, which has been approved by most regulators in recent years. And as you can guess, it works by targeting the PCSK9 mRNA, leading to a reduction then in this protein. And when used, you go from a situation where you have too much LDLR degradation to a situation like this here on the bottom, where you have less of this happening, and as a result, you get normal levels of functional LDLR and less harmful levels of LDL in the blood, even though these patients are heterozygous for a hyperactive PCSK9 mutation. This, of course, means that it should also be a benefit to patients with heterozygous dominant loss of function mutations for LDLR, in the example described earlier. Because although the PCSK9 protein is not overactive in these patients, if you can decrease PCSK9 driven natural degradation of functional LDLRs in these patients by inhibiting their own natural PCSK9, it seems only natural that, at least in heterozygous carriers of this loss of function LDLR gene, they may benefit from this also, and indeed they do. This may explain why there's no real big movement towards LDR-based mRNA protein replacement therapy for these individuals. Inclisiran is approved and suitable for counteracting FH caused by both loss of function and gain of function mutations in these different genes. So it looks like it's going to be a huge success, and in fact, sales are expected to reach 2.8 billion by 2028. So it's a remarkable example of the real clinical relevance of nucleic acid therapy. But for these kinds of dominant mutations where you need to silence a toxic gain of function or dominant negative gene, don't forget that sometimes the normal versions of these genes can also exhibit haploinsufficiency, like in this example here. So, in some cases, after silencing a mutant gene through antisense oligonucleotides, you may still have the problem that a single normal copy might not be enough to produce high enough levels of normal functioning protein. So, simply silencing a harmful gene alone may not always be a solution. So, in these situations, it may be best to combine gene silencing with gene replacement. So, after silencing, then protein activity is restored in a therapeutically beneficial manner. But this involves using two therapeutic molecules. So, as an alternative, rather than silencing a gene, in some cases it may actually be possible to use a single molecule to convert a harmful gene into a less harmful and somewhat functional version. And through this approach, you might be able to compensate for this haploinsufficiency. And this may be achieved by using a different type of antisense oligonucleotide known as a splice switching antisense oligonucleotide. And these work by exploiting the fact that in cells, RNA is naturally transcribed as a pre mRNA first, containing regions called introns. And these are removed in a process called splicing, leaving us with a fully processed mRNA that only contains the protein coding regions called exons, and with a fully unbroken protein coding sequence. However, it's possible to inhibit the inclusion of some problematic or mutated exons in the final mRNA by targeting the pre mRNA at the splice sites using one of these splice switching antisense oligonucleotides, leading to a slightly different version of the mRNA and, of course, then a different version of the protein that hopefully doesn't exhibit the same toxic gain of function or dominant negative activity. But instead, the hope here is that it functions more like the normal version of the protein, or that it at least retains enough of the original function and it can work alongside the correctly functioning protein to make up for this haploinsufficiency. So the idea here is that it's the combined levels of the good protein and the new somewhat functional protein that lead to the therapeutic effect. And in terms of clinical uses, in recent years there has been a lot of activity with splice switching oligos to treat DMD, for example, which is a muscular disease. And for this, there has been at least four splice switching ASOs filed with the FDA for review. Two in 2016, which act by skipping exon 51 in the DMD gene, and two others filed later, which act by skipping exon 53 in the same gene. One of these was rejected, but even though the others were approved, there really appears to be very little clear evidence of any therapeutic effect against DMD with these ASOs or splice switching ASOs. So at the moment, these are quite controversial, so no harm being aware of that. A better example might be nusinersen, which is a splice switching ASO against spinal muscular atrophy. But sometimes when you have a toxic gain of function or a dominant negative gene and you need to silence it, you might not be able to do so. Maybe it's not possible to target the mutant gene via its messenger RNA without also impacting the normal gene, or maybe the silencing itself comes with a lot of off target toxicity, like with some gapmers that have been used to treat FH. Also, maybe with the alternative, which is splice switching, it might not be possible to skip exons in a way that results in the generation of a functional protein, which may help you overcome any haploinsufficiency. So, if any of these scenarios exist, antisense oligonucleotides are not really an option for silencing or for combined gene silencing and gene replacement approach. However, rather than working at a transcription level, it's also possible to counteract a problematic gene by modifying the genome in a way that removes or disrupts the problematic gene. This is referred to as gene knockout. And the problematic gene can also be replaced in the genome by a correctly functioning version, in theory. So, as some of you might be aware, this can be done through the use of CRISPR Cas9, which is a technology that's gained a lot of headlines over the last few years, the development of which led to a Nobel Prize for Dr. Jennifer Doudna and Emmanuel Charpentier, one of whom I actually got to meet recently when they were over here in Liège. And this is a technology that has really made it a lot easier for us to make precise changes in genomes. And it's something we've even used ourselves here in our lab to knock out important immune genes in zebrafish as part of some of our publications. And it's something that really has revolutionized what we can do in terms of fundamental research. And today I'll introduce its application in terms of nucleic acid therapy. But first, let's take a look at how it works. So, in principle, the way it works is not too dissimilar from the siRNAs that I described earlier. But in this case, a CRISPR guide RNA is incorporated into what is referred to as the Cas9 protein. And this guide RNA is designed to undergo base pairing with regions of DNA that you want to cut. And after the guide RNA binds, the Cas9 protein makes a double stranded break in the DNA. But it's important to remember that you can't just cut anywhere. The cut will only occur within a few bases of what's called or what's referred to as a PAM sequence, which is a small three-base sequence which can start with any nucleobase followed by two guanine nucleobases. So basically, you need to have a PAM sequence within a few bases of your cut site. This initiates, broadly speaking, one of two natural DNA repair mechanisms in cells. In cells that are not dividing, this will typically take the form of non-homologous end joining, which is a quick but haphazard repair mechanism, and it typically results in the random insertion or deletion of bases at a cut site after the cuts are repaired. And of course, as you remember from earlier on in this lecture, in protein coding genes, any kind of insertion or deletion like this leads to a frame shift, leading to a completely different protein being translated. Or in this context, it effectively equates to the loss of a dominant toxic gain of function or dominant negative mutant gene that causes a disease. Alternatively, instead of this DNA strand breaking and haphazard repair, by supplying an external DNA sequence, it's possible to get this incorporated into the site where you have made the double-stranded break. And this occurs due to a process that's called homology-directed repair. And it can occur to a much greater extent in dividing cells. In this case, the insertion of the desired DNA sequence at the double-stranded break sites relies on sequence homology with regions flanking the double-stranded break site. In other words, the DNA sequences flanking the double-stranded break site and the sequences flanking the new gene need to be the same. So it's just a matter of designing your DNA insert to have these sequences. In either case, the initial double-stranded break can be achieved by delivering, for example, DNA with the genes for the Cas9 protein and the guide RNA, which can then be transcribed in the cell. Or you can just directly deliver the Cas9 messenger RNA and the guide RNA. Although in this case, the guide RNA needs to be stabilized as it does tend to degrade a lot in the cell while the Cas9 is being translated in the cell. And of course, this direct delivery of RNA is more desirable because once the genome changes are made, you don't want the guide RNA or the Cas9 protein hanging around, potentially editing other parts of the genome. So once the RNA degrades, any further genome editing ends. Whereas with DNA delivery, you have new guide RNA and Cas9 being continuously made all the time from the DNA that was delivered. So you have more of a chance of accumulating off-target genome modifications using this approach. But in any case, after the double-stranded DNA break, the next step can be either gene disruption or gene replacement. And of course, gene disruption is much less complex. So it's no surprise that this kind of an approach has got to the clinic first, being utilized here in the first approved CRISPR-based therapy called Casgevy, which will be used in the treatment of sickle cell disease. And as this is a blood disease, any in vivo delivery issues can be largely circumvented by delivering the therapeutic molecules directly to the blood stem cells that have been removed from patients and then returning the treated cells back to patients via infusion after modification. And although sickle cell disease is caused by a mutation in the beta globin gene, it's not actually a gain of function or a dominant negative mutation. It's important to point that out. And also, instead of altering the beta globin gene, they disrupted the expression of another gene, leading to the increased activation of a third gene called gamma globin that is somewhat functionally similar to beta globin. And this is where the therapeutic impact comes from. So this kind of an approach is much less complex than gene replacement using CRISPR-Cas9, so it's no surprise that most CRISPR-based therapies currently in phase 1 to 3 clinical trials involve knockout approaches like this. And like in the case of sickle cell disease, the majority of these therapies are delivered outside the body also. But it does open the door for eventual progress towards CRISPR-based gene replacement, because let's not forget, in other cases, you may need to replace a toxic gene with a normal gene, as described earlier. But the approval of Casgevy, even though it relies on gene disruption, is still an important milestone, of course, that will lead us someday, I would expect, towards CRISPR-Cas9 gene replacement. So it'll be very interesting to see what happens here over the next year or two. But remember, as an alternative to modifying genes in this way by making double-stranded breaks in the DNA and then inserting or deleting chunks of code, it's now actually becoming possible to make direct changes to the code itself through base editing and prime editing techniques. But I'll talk about this in a different lecture. But for now, just to finish up, I prepared a second table to summarize what we talked about again today. Again, I'll send this on to you. Like the other table, there's a lot of text here, so I won't go through it in any detail, but it just summarizes what was outlined here today in terms of what kind of nucleic acid therapies are optimum, or in some cases not optimum for different types of mutations and inheritance pattern combinations. For example, recessive, loss of function, gain of function, dominant negative mutations existing in either a heterozygous or homozygous state, also with some considerations regarding haploinsufficiency as well. And just for completeness, I've also added X-linked inheritance patterns here too at the end. And each modality is also color-coded here as well, just to indicate how suitable it might be in each scenario, and just to make things a little bit easier to digest. But it's by no means a complete summary of the entire field, of course. It's just the basics, and there's always more than one or two ways to get things done. And in biology, as some of you will know, there's always going to be exceptions to the rules. But I think it's certainly enough to give you a good introduction to the area. And for sure, don't hesitate to get back to me if you have any questions on it, or if I've left some typos in there, which is, of course, possible. But that's all from me for today. I hope you enjoyed that introduction into the clinical significance of nucleic acid therapy and how it differs from other types of therapy, the various modes of action in vivo, and the kinds of things that are considered when we're trying to tackle inherited conditions using nucleic acid therapeutics. And importantly, I hope you can also see as well why there's so much potential surrounding it. And although I focused on inherited conditions, as I mentioned earlier, nucleic acid therapy can be used to also counteract non-inherited conditions or to supplement existing proteins or introduce new therapeutic proteins such as antibodies. And of course, this is all in addition to its use, the use of nucleic acid-based vaccination against infectious diseases. And in the next lecture, we'll continue on this theme with more of a focus on nucleic acid therapy in the context of cancer. As I said earlier, I think it deserves its own separate lecture. There'll be a bit of immunology in it, but not too much. So until next time, take care, and I'll see you all soon.